again, those questions that seem to be several people did not get right on exams or on homework. What I want you to do is, and I'll go through that real quickly, how these are on this list. But then I'm going to want you to vote for your top, the top thing you want to discuss today. So we'll start with the one that most people want to hear and go down from there. So at the beginning is something that you had as a homework problem, and that is if you're doing dideoxy sequencing, where will the bands be found on the gel as you add nucleotides to the primer? Something like only 60% of the class got that correct. That was a very early homework. You may have figured it out by now. Another one that was low in early homework was understanding how to predict a given amino acid sequence based on whether the code, the genetic code is 2 nucleotides or 3 nucleotides or 4 nucleotides. And this was the mechanism by which the genetic code was ultimately deciphered, that it was a triple code. So again, we can go over that. Binomial expansion is something we had on this last midterm, and 27% of the class got full credit for that. Almost 50% of you got at least 70% of it right, but that means 50% of you didn't. So that's why I put that up there. Chi-squared, primarily I think an issue of understanding how you relate chi-squared to probability and what we consider rejecting or accepting the null hypothesis. Genetic complementation was another place where actually generally you did very well, but I have a feeling you didn't quite know what it was you were looking at. You couldn't name it. Then RNAi, someone in the class wanted to go over that again, how it works. Chromatin remodeling and chromatin modification, again, a request from the students. The request from students how to screen a cDNA library for a gene of interest, how to look at FLTs, and then the Southern technique. So, you've had a minute to look at this, and I'm going to just go down the list and get votes, okay? What we'll try to do is just look for a huge, so vote for only one thing to begin with, okay? Look at your top priority. So, dideoxy sequencing. Deciphering the genetic code. Binomial expansion. Okay, I've got 10 or 15 for that. Chi-squared. Okay. Genetic complementation. Okay, RNAi.
5%.
Chromatin remodeling and modification.
I can never go there and really come up with that.
I'm already getting, oh, sorry, I don't have a pen. Does anyone have a pencil they can hand me or a pen? Thank you very much. You still have that. All right, so again, was it binomial expansion was about 15? The big 10 was for what? Okay. 5. Okay, and then chi-squared, we had chi-squared, just a few, right? RNAi. Well, that was another good one. Chromatin remodeling. Okay. How to screen a cDNA library. FLTs. And Southern blot. Okay, so we're going to start with the genetic complementation.
There's actually an Eastern block and Eastern South block.
Okay, I just have one slide on this. Yeah. Can you use the microphone? Is that possible? Well. Okay, can you hear me through that?
Are you posting these?
Yes, I'll post.
Okay.
Okay, so the main thing about genetic complementation is what you look for is that you have mutations, both of which give you a similar mutant phenotype. And in this particular question that was asked on the exam, one thing you want to look at very carefully is it says pure breeding. What does pure breeding mean?
Homozygous?
Exactly, homozygous for the trait. So that all of its progeny are going to have the same alleles, that's all that can be transmitted, is the same mutant allele. So these were two homozygous mutant strains, and they had a mutant phenotype, which gave them a sphere shape. And then you make them, and you suddenly get wild type. That's genetic complementation. So as soon as you see that, mutant times mutant gives you wild type, and someone asks you, what does that demonstrate? Genetic complementation. They're complementing one another's problems, okay? One has a problem, the other has a problem, but they complement each other's problems to come up with a wild type. So, so another thing that's pointed out here is that if you got mutant times mutant and you got mutant, you actually wouldn't know for sure whether they were mutations in the same gene or not. It's possible that they're dominant mutations, okay? But whenever you see mutant times mutant gives you wild type, they must be in different genes. The mutations must be in different genes. That's the only way you can get complementation. So what it means is that one is mutant in gene 1, the other is mutant in gene 2. In this example, we were actually given A's and B's. So one's mutant in gene A, the other one's mutant in gene B. Both mutations are recessive. So when you make an F1, your big A, little a, and your big B, little b, where the little a and the little b is the mutant. And since the wild type is dominant, you end up seeing wild type. So you can tell all of that just by saying, I took two sphere-shaped things, mated them, and I got wild type. Okay. So that's where it says mutant times mutant equals wild type is called genetic complementation. Okay. And then I just asked you, could these be dominant mutations? No, because if they were dominant and they were in two different genes, then you would have dominant, that's where I'm telling you, it's possible for them to be in two different genes, okay, if you still get mutant when you mate them. but they cannot be the same gene if you see complementation. Okay? So the bottom line is, if you see two mutants to give you wild type, it tells you right away that those two mutations are in different genes. Okay? Then the other part of this question asks you, to answer the second part of this question was when you made these two, the F1s themselves to each other. Okay, so you take this wild type times wild type, but it's where you've gotten a mutant times a mutant gave you wild type. That wild type you should now realize is big A, little a, big B, little b. If you make an F2 generation from those, this is where you, at this point in your training, should immediately know this is a dihybrid chromes, right? These are, because it's two different genes. We've already said it's two different genes. So what we expect is a phenotypic ratio of 9 to 3 to 3 to 1. That's just basically, you can draw out the Punnett square, okay? Or you can do the other kind of diagram. But ultimately, you should be at this point so familiar with dihybrid crosses that you know right away that you're going to have 9 that have are big A and either big A or little a, big B and little b. And then you're going to have 3 that are homozygous little b and heterozygous or homozygous for big A. And you're going to have 3 that are homozygous mutant for A, and then heterozygous or homozygous big B. And then you're going to have 1. Okay, so these are the predicted ratios of the phenotypes you'll see. When you don't see the predicted ratios, what does it tell you? Epistasis. Okay, it's saying that what you have at one gene is influencing the expression of the other. Okay, so we went through a million examples of epistasis. What I'm not going to ask you is to tell me dominant versus recessive versus this versus that kind of epistasis, but I do want you to recognize epistasis. And the best way to recognize epistasis is a deviation from the 9-3-3-1 ratio. So in this case, it was 9-6-1 instead of 9-3-3-1. Okay? Questions? That's the genetic complementation. Okay, so the next highest on the thing was RNAi. So RNAi refers to RNA interference in gene expression. Okay. But that doesn't mean that it is literally affecting expression that is transcription of the gene. It means that you're affecting gene products expression through RNA. And in fact, RNAi acts through both transcriptionally and post-transcriptionally. The main things I want you to know about RNAi is that there are 2 types of RNA molecules that are involved in RNAi. We have microRNA and we have small interfering RNA. So the microRNA is RNA that is made from a DNA sequence that has its own palindrome. And remember, a palindrome is where the DNA sequence is flipped around, okay, and relative. So you're reading in one direction. Well, actually, we show with restriction enzymes, right? So I think you can write this down because I can't write it down. I think GAATTC is the ECHO R1 restriction recognition sequence. If you write that as a double-stranded sequence, then reading the top strand from left to right is GAATTC, and reading the lower strand from right to left is GAATTC. That's a palindrome, okay? So that's what that's kind of, this is a case where the RNA strand, so in this case, it's the upper strand that can be read, that can fold upon itself, okay? So that you make the RNA, and you've got this long piece of RNA, but there's a sequence out here that can base pair with the sequence over here, all right? And so it makes a stem. We call the stem is where they pair with each other. I'm going to break my wrist, okay? So they are pairing. Where they pair is the stem, and however big that loop is, that's a loop. So it's a stem loop structure. That's a microRNA. Small interfering RNA is a situation where you've got a region of the DNA where you're transcribing in one direction, which means if I'm looking at you and I'm going this way, this is the way we usually have it, okay? The RNA is making a copy of the lower strand, right? So it's transcribing that direction. But also, there's transcription occurring in this direction. So when I'm going in this direction, the lower strand is my template. But when I'm going in this direction, the upper strand is my template, right? Again, this is something, please know by the time you leave this class, all right? You really need to know about transcription and which strand is template and which one is the mRNA. Okay, so the coding strand. So there are some regions in the DNA we've discovered are actually being transcribed in both directions. So if you make those two RNAs, they're going to be complementary. So they're going to make double-stranded RNA. Why does that matter? Well, it turns out that we have evolved and a mechanism for dealing with double-stranded RNA is generally considered a problem because it's not well, at first we thought it was just a response to infection, that we learned how to get rid of RNA viruses and things of that sort. But now we realize this is a naturally occurring mechanism for regulating gene expression. So whether you get what you're seeing here is bidirectional transcription, which is giving you that small interfering RNA, which gives you a double-stranded RNA sequence. Over here is demonstrated a case of microRNA, where it's folding upon itself, and that little spinning thing at the bottom is supposed to indicate that it's base pairing, making a stem, and then a loop. Both of these get recognized by a protein called dicer. And dicer then cuts the double-stranded RNA into discrete pieces of roughly 21 to 25 base pairs. Once it's been cut, it is recognized by a complex of proteins called RISC. That is RNA-induced silencing complex. Okay? So that's not one protein. Dicer is one protein, but RISC is a multi-protein complex. When it finds that RNA, there is the two strands come apart. One of them is then deemed the passenger strand and is thrown out of the complex. And the other one is called the guide strand, meaning it's going to be used to guide the RISC complex towards a complementary sequence. Okay? So the guide strand is the thing that's going to base pair with the target. And when it does, it's bringing the RISC complex with it. In most cases, we're finding that RISC does this. It guides, the RNA guide strand brings the RISC complex to messenger RNAs to which the RNA is complementary. And so now what you've got is RISC binding to a messenger RNA, and it reads it. And in that way, knocks out expression of the gene that made that mRNA. Okay. It can also act just plain block translation. So we originally thought that that is actually how it worked altogether, was that if you got double-stranded RNA, a ribosome cannot bind to double-stranded RNA, so it cannot be translated. You know all about how RNA sits in a ribosome, right? So double-stranded RNA will not get translated by the ribosome. So that's another way to stop expression. We ultimately learned that that's not the only thing that was going on. In fact, we were getting degradation of the messenger. Today, we use this as a technique. We make our own silencer, our small interfering RNAs, or we develop our own hairpin stem loop structure RNA to put into cells to try to turn off genes whose function we want to study. Okay, so if we just throw a bunch of this into a cell that's expressing a particular protein we're interested in, we can keep that cell from making that protein using this technology. All we have to do is provide the double-stranded RNA, and the cell will do the rest. What has been discovered more recently is that some such guide strands actually go all the way back to the DNA and block transcription by forming some kind of tri-strand structure. That's not as well worked out as the others. But the indication is that in some cases you actually get transcriptional arrest. So you don't have any mRNA either because it's not being allowed to be made, that is, be transcribed from the DNA, or because it's being degraded, or it's still there, but it can't be translated. So these are the various ways that this RNA interference works. Questions? Yeah.
I think in the book on that section, it says that it gets diced to about that length, 21 to 25, and then it folds over to a size of typically containing 65 to 70 nucleotides. Or is that the other way around? Looks like it gets longer or not?
No, it doesn't get longer.
Okay.
Yeah. So, I mean, it's a double-stranded thing. So you've got 21 to 25 base pairs that are in the siRNA. So you want, when we synthesize them, we want to have complementary sequences of about 21 to 25 base pairs in order to make these things work. Yeah.
Do those like RNA interfering genes, are they considered genes, and do they have promoters?
Okay, so they do have promoters. This is a very active area of research, is trying to figure out where they are and what they're doing. So one of the things with RNA-seq, where people doing copious amounts of sequencing of RNA from cells, they're discovering these things that don't seem to have any coding capacity. And then they're going back and looking at the DNA to try to figure out why they're made and under what kind of control. So I don't think that I can give you a global answer as to how they're expressed. Yeah.
Maybe I'm confused. Right. Are the siRNA and miRNA purely accidental, or are they intentional?
No, so that's what I was saying, is that we believe they are intentional.
Right.
Okay. So he was asking, are they accidental that we make these things, that our cells make these things? Initially, we thought that dicer was in our cells because it was a defense against viral infections with RNA viruses. But as people then isolated dicer and then discovered risk, they discovered that in fact our cells are making such double-stranded RNAs routinely. Okay, so they seem to be a natural component of gene regulation. I can't tell you for any particular, every system is now going back to look to see whether RNAi is a part of the regulation of the gene system that they're looking at. Okay, so for the longest time, you know, one of the things that we were talking about earlier, I worked in immunoglobulin genes. You know, they're all, the heavy chain genes are all in a tandem array, all pointed in the same direction. And then you start to find that within intronic sequences, there's some transcripts going the other way. So now people are trying to figure out, do they have a function? Okay, but they would form double-stranded RNA for the transcripts. Okay. So, so then I have how to screen a cDNA library for a gene of interest. So this is where I was saying that whether you go directly to try to find your gene in the genome or you make a cDNA library is influenced by what you're trying to do and what you already know. Right now, people, for example, will take two different cell types. Say you have a cell and then you treat it with a drug and you want to know what the drug has done to the cell. Many people will now sequence all the RNA that's made by the cell before the drug was added, and then sequence all the RNA that is made by the cell after the drug was added, and look for changes. In that case, are they sequencing RNA? No, in fact, we don't sequence RNA. They're making cDNA, okay, and using the cDNA to get sequence, okay? But more generally, I think I gave you the example that's shown on this slide. When we wanted to clone the immunoglobulin genes, and it was very huge desire among the genetics and immunological community to figure out how those genes worked, because they seem to be doing such bizarre things. And what was done was to start with a cDNA library because we had cells that we knew made immunoglobulin. If they make that protein, they must be making the message. And if we can get the message, it will give us basically the DNA sequence, right? It'll give us part of the DNA sequence. It's going to tell us what is the actual sequence of the mRNA making the protein. This brings me back to some questions you got on exams or homework, and that is, if I have the amino acid sequence for a protein, from that, can I tell what the mRNA sequence is that gives rise to that protein? No, I can't, because there is more than one codon that can encode a single amino acid. Okay, so I can't tell which codon was used to make that alanine, or which codon was used to make that leucine. Okay, so I can't just go in the protein and then look for a particular DNA sequence. So I make a cDNA library from a cell that's making a lot of this protein, in the hopes that in that library there is a clone, and we talked about making the cDNA library, and we put it into a vector, and we can put it into bacteria, for example, into bacterial plasmid, and we can clone each one of the E. coli, each one carrying a different copy of a message. And that's what we mean by the library. But now I want to figure out which one of those clones encodes the protein I'm interested in. So what you see here is that I really can only do that if I have some way to detect the product. Okay? Because I'm not going to know whether the clone encodes the protein if I don't know how to detect the protein when it's there. So if I'm gonna, if I'm gonna, well, actually, there's some techniques that allow you not to know that, but we'll get to that in just a minute. So in general, what I want to have is some way of detecting my protein. All right, so the most common thing to do is to clone your cDNAs, and in fact now these days people only make their cDNA libraries in expression vectors. And now you guys know what an expression vector is. It's a vector that's going to provide the promoter, an enhancer, a transcription, a polyadenylation site, okay? Because remember the cDNA does not have promoter, it does not have an enhancer, it does not even have the poly-A recognition site. It has poly-A sequence. But remember that recognition site is internal to that, okay? And that's where it got cleaved. Well, it's not where it got cleaved. It got cleaved downstream of that and then polyadenylated. So at any rate, we generally will end up putting a polyadenylation site in the expression vector. So you can either put it into a prokaryotic or a eukaryotic expression vector. And you should know what the difference is between the two, okay? It has to do with the regulatory sequences. Regulatory sequences, meaning promoters and enhancers that work in bacteria, do not work in eukaryotes and vice versa. Okay, so now what you're going to do is you're going to take your cDNA library and if you put it into prokaryotic expression vector, say bacteria, then one thing that you can do is simply put a piece of membrane down on all the colonies, the bacterial colonies. When you pick the membrane back up, you've got little pieces of colony stuck all over the filter. You can, in fact, treat that to lyse the bacteria that's on that membrane, and all the proteins that are being made by that bacteria will now be associated with that membrane. Now you can incubate that membrane in a solution that has antibody that recognizes the protein of interest. So if the bacteria are making that protein, then the antibody will bind to just that spot on the membrane where that bacteria was. Okay? And we have detection mechanisms, say, again, we can link an enzyme to the antibody and then add the substrate for the enzyme, which will give you, say, a blue color, like you guys were talking about in your lab. So I can end up seeing that the membrane has a little blue spot, or 2 or 3 blue spots, and then I align that membrane back with that petri dish where I had all of my colonies, and I say, oh, that spot is right where that colony is. So I'm going to pick up that colony, and I'm going to grow it up to large amounts, and I'm going to lyse the cells, and again, test, is that cell making this protein? So that's one way we screen for the product, for the cDNA. We're looking for the product, okay? In some cases, you do it in eukaryotic cells instead of in bacteria, okay, which you can do. And it's really the same sort of situation. Now, there are cases where you don't need to know the actual protein that's involved. Instead, what you're trying to do is find the wild type form of a gene product that you have mutated. Okay. So, an example I gave you in lecture before. We use yeast, okay? So here's an example of yeast that carry a mutation, turns out to be a mutation in a cell cycle gene, okay? And it's a temperature-sensitive mutation, and so that means that these yeast, you can grow to high numbers even though they're mutants in cell cycle, which is a problem, right? If you can't replicate, you can't keep a strain growing if it has a mutation that keeps it from growing. But if it's a temperature-sensitive mutation, then if you grow it at the permissive temperature, it can grow, all right? It's only when you shift it to the non-permissive temperature that the mutation is revealed and the cells cannot divide, okay? So that's the situation here, where we have a yeast strain that has a mutation in a gene, and we want to know what is that gene product, okay? So we take those yeast cells and we transfect them with flat expression vectors that work in yeast. Yeast are eukaryotes, okay? So we transfect them with the library, and then we take, and again, we can use like you guys used. You used a positive selection, like the drug, okay? So we can include a positive selection marker on that plasmid so that only yeast that have picked up plasmid are able to survive at the permissive temperature. But generally, all the cells will survive as long as they're at the permissive temperature. And now, again, we can blot down on that plate of yeast with a membrane or with a piece of, in the old days you used velvet, okay? But you're just physically picking up a copy of all the yeast colonies off of the plate, and you're setting them down on another plate, so you have a replica plate. And here you now take that replica plate and put it at the non-permissive temperature. What should happen? What should happen to most of the yeast colonies you've picked up and put in this replica plate and now put at non-permissive temperature? Most of them should die, because we started with a population of yeast cells that are all mutants in this one gene, okay? This temperature-sensitive mutation. But if I see a colony growing up at that non-permissive temperature, it's an indication it's picked up a plasmid that can cure, that can complement that mutation. So just like we did genetic complementation, 2 mutants make a wild type, this is a case where we have a mutant, but we add a plasmid, and the plasmid now takes care of the problem, okay? So now we're going to isolate the plasmid from that one colony and try it again and see that now every yeast that picks it up is now able to grow at the permissive temperature, at the non-permissive temperature, okay? And therefore, we have thought that we are likely to have the gene that was mutated. Now, one thing I pointed out to you guys, all right, is that there's such a thing as suppressor mutations, right? So it's possible we picked up something that, when overexpressed, it can complement the mutation in the gene of interest. But in general, usually what we're picking up are true genes in that way. So in that particular case, I don't know. I can call it CDC2, but I don't necessarily know anything about the protein that gene encodes. But I'll find out about it by cloning a cDNA that can cure that mutation. Okay, questions? Okay. Okay, so the next thing I see here is chromatin remodeling. So So again, the big message to you about chromatin is that we've learned about cis-acting regulatory sequences, which are promoters, proximal regulatory regions, meaning regions that are very close to the promoter, and then these regulatory sequences like enhancers and locus control regions and silencers, which are really defined by the fact that they can lie a great distance away from the promoter of the gene they regulate, and they in fact can be put in the DNA in either orientation and still work. Okay, whereas the promoter, you have to have a Tata box in the, it is in a particular site downstream of which transcription starts. So you can't flip a promoter. It has to stay in its natural orientation to drive the expression of the gene it regulates. Enhancers, silencers, LCRs can be at a distance and can be in either orientation and do their job. But that's only part of what's going on, and in fact, part of how they work to regulate genes is to influence chromatin around the gene. And so that's why we're talking about chromatin. It's another level of gene regulation, and many people refer to it as epigenetics, because you can change whether a gene is expressed or not by changing anything about the DNA, but rather changing the way it's wrapped around nucleosomes, okay? So it's not something that actually changes the genetic material. That's what is meant by epigenetics in this case. So we have basically two different kinds of things that happen. One is we call chromatin remodeling, which is talking about moving nucleosomes to get out of the way so that regulatory transacting factors, that's those transcription factors and such, that want to come and bind to those cis-acting regulatory elements. Okay, so the DNA is a regulatory element, whereas transcription factors are proteins that come to lie down, sit down on the DNA to do the regulation. But they can't get in there if the DNA is all coiled up with no nucleosomes. So we know about several different systems that can remodel and move nucleosomes out of the way. And the classic ones and the ones I want you to know about are the SWE-SNF family. There's I-SWE, which is over here in blue, which works in the capacity of aiding in the winding of nucleosomes around the DNA to form nice, tightly packed, ordered array of nucleosomes. So it's a wrapping things up kind of complex. But SWE-SNF are proteins that are involved in pushing nucleosomes out of the way. And so the two ways that they do that is either to just kick the nucleosome out, and that's what's seen here in the middle, nucleosome ejection. Or, so you see that we have 5 nucleosomes, and one was completely packed around the Tata box so that this promoter cannot work. And this SWE-SNF can throw that nucleosome out of the way, making now that Tata box available for activator protein, such as the Tata binding protein. Or instead, it's shown in the top, instead of getting rid of the nucleosome, it slides it away, out of the way. Okay, so it just slides down the way. So you still have 5 nucleosomes as shown in this picture, but one has been moved out of the way. And then the other way in which they can regulate how the nucleosome packing works is to put a modified histone protein, H2A, .Z. It's a modified form of the H2A component of the nucleosome, and it is much less tightly bound to the DNA, and it makes it a more flexible nucleosome that, again, can be taken out of the way. So this is what we mean by chromatin remodeling. Okay. The other major kind of regulation of chromatin is chromatin modification. And what we mean by modification is really the modification of histone proteins within the nucleosome. Okay. So your text points out the fact that the way that this whole system works is there has to be a mark itself, and in this case it's going to be acetylation or methylation. Those are really the two major types of marks that we see in chromatin. And then we have to have an enzyme that adds marks, which they call writers. And then you have to have enzymes that get rid of marks, which they call erasers. Okay? It's all pretty simple, right? But this regulatory process is not very simple at all. And what you're learning here is just to get an overall picture of what we're learning about how we regulate gene expression through these modifications. The summary information that I want you to take home is that in euchromatin, where the nucleosomes are not tightly packed and those transcription factors can get in and actually act on genes, that's euchromatin, EU chromatin. Okay? In those places, you see hyperacetylated histones, meaning they have acetyl groups added to the tails of the histone proteins. And in DNA that is heterochromatin, you don't see very much acetylation at all. You see a lot of methylation. So that's what you're seeing here, is the heterochromatin has a lot of methyl groups, and the euchromatin has more acetylated groups. But it's not absolute. Some types of methylation are associated with active chromatin, meaning euchromatin. Okay? What I wanted you to know about the nomenclature is what this refers to. This refers to histone 3, okay? Lysine number 9 is acetylated, okay? And this is histone 3. Lysine number 27 is trimethylated. Okay, so that's what that means. It's just telling you about which histone protein is acetylated or methylated and on what amino acid residue. And in fact, what you'll see is that methylation on H3K9, okay, is associated with heterochromatin, but H3K4 methylation is associated with euchromatin. It turns out that it's the complex of, how many you have of this kind and how many you have of this other kind that ultimately determines what happens to the gene that's underlying all of this chromatin. Again, this is, you could take an entire course on chromatin modifications.
Although it's methylated, you said it's a B chromatin?
Yes. So there are some methylations on histones that are associated with euchromatin. And so when I say that overall, you're going to see more methylation in heterochromatin and more acetylation in euchromatin, it's not absolute. And in fact, there are you will not find this kind of methylation. You will usually not find this kind of methylation in euchromatin. But again, it's a matter of level as well. So, it's like a shifting level from one to the other. Yeah.
Here we talked about methylation. What about the DNA itself?
So we didn't talk in this class about methylation of the DNA. But in fact, that was the first thing that was discovered about silenced genes, is that the DNA itself gets methylated in silenced genes. I'm not going to test you on that because we didn't cover it in the course. But it is something good for you to know. And that is, when we first started trying to figure out why is this gene expressed in this one tissue when it's not expressed in this other tissue. We used restriction enzymes, you know, as we've already done, to look at genes and where they lay and all that kind of stuff. And it turns out there's some restriction enzymes that are sensitive to methylated DNA. And because of that, we can ask, well, if this enzyme is sensitive to methylated DNA, is it able to cut this gene in this cell type? And will it cut this gene in this other cell type? And we found that some of those enzymes would cut the DNA in one cell type and not the other, which implied that in the other where it wouldn't cut, that DNA was methylated. Okay, so DNA methylation also happens. Okay, so I think the next thing we have on here is the RFLPs. So RFLPs actually been used in for a lot of different purposes. And I won't go into the history of the first really cute way they were used with immunoglobulin genes. But the bottom line is that we can construct a genetic map using known phenotypic mutations, right? You guys had all these lectures talking about looking at a couple of different traits and then looking to see if those traits are linked or not linked. And if they are linked, what's the recombination distance between them? And as a result, you can ultimately end up with a genetic map of a whole chromosome that tells you this gene is next to this gene, is next to that gene, is next to this other gene, and they're separated by this number of recombination centimorgans. Okay. Well, as soon as we started to realize that DNA could be looked at directly for allelic differences, then it became obvious you could generate a map of these differences in DNA sequence. In particular, for example, restriction fragment lengths, or even single nucleotide changes. Okay? So we get to this because we can have, say, a gene, we want to understand, we know it's important to something that we care about. And we know that if we mutate this gene, it has a really important effect on this thing that we're interested in. But we have no idea where to find it, okay, in the DNA. And in fact, these DNA markers can help us get to that. And so, this just demonstrates to you what I mean, and that is that once I have on the left over here is the on the left, far left shows you centimorgans, and just to the right of it on that same, this is chromosome 5. Then you have a series of genes that have been identified by phenotypic mutations, and you've just done the right kind of reading and so forth to map them relative to one another and know how far each one is from the next. Many years devoted to getting that map. Okay. In this second line is what has been generated in many fewer years once we had access to the DNA. Again, taking a region of DNA where we find that, for example, you get using a piece of DNA, just a random piece of DNA that we ultimately learn lies at this far end of this chromosome, which we'll call 4.1, okay? When we use that as probe, we, in one strain of fly, we get a 5 kilobase fragment of FOR1, and in another strain, we get a 3 kilobase fragment. And that it behaves, those are genetically inherited. If you make a heterozygote, you have both a 3 and a 5 kilobase fragment, and you can follow them through various matings. We get another piece of DNA, and it gives us 2 different restriction fragments from those 2 different strains of flies, and we follow those 2 different alleles. And ultimately, we figure out, oh, they're linked, okay? And you know how to find out if they're linked by your matings, okay? But they do recombine at some frequency, and the recombination frequency tells me how far they are from one another, okay? So I can generate this entire map of DNA, again using genetics, but using molecular probes as I do the genetics. But now what I have is I have the opportunity to clone all the DNA in the flies. Okay? I'm using the fly as an example here. And we already talked about that. That is, we take the whole genome and we cut it with restriction enzyme. We do those partial digests. We clone big segments into vectors, cosmids, and so that we have a library of the entire genome. Well, now I can take that probe that I know sees that polymorphic marker 4.1, and I can use that as a DNA probe to search that library for its complementary sequence. And so I can clone out a 100 kilobase fragment of DNA that includes that piece, because I find that that piece will hybridize to the cosmic plum that has that big piece in it. Okay? Are you following me? No. Okay. So I take the whole genome, right? And remember we said what we do is a partial restriction digest. We don't want to do a full digest because we may cut our gene of interest. So we do these partial digests, which means that each vector, so here we're seeing a back vector, all right? We're ligating a big piece of DNA to a back. That's just one plum. And then there'll be another big piece of DNA going into another back plum. Back is just a kind of plasmid, okay, that accepts a huge amount of DNA. It stands for bacterial artificial chromosome. But what happens is you transform E. coli, and just like we said for the cDNA library, every one of those colonies has an individual big piece of DNA. So now I'm going to do the same old thing. I'm going to put a membrane on top of the bacteria. I'm going to pick it up. I am again going to denature so that I lyse the bacteria. But now what I'm going to be paying attention to is the DNA that is bound to my filter. Okay? Before we were talking about protein. Okay? But also we're picking up, when we pick up the E. coli, we're also getting DNA, all the DNA that's in the E. coli, including multiple copies of that back. So each one of these is containing a different piece of the genome. So I have this filter. I have dots of DNA all over this filter. I put it into a solution that has radioactively labeled DNA from that 4.1 region. Okay? That thing where I found that there was a polymorphism, 4.1. So I have a little DNA that's from this region that I know is polymorphic. The radioactive DNA, actually we put in a probe that's double-stranded, but we put it in conditions where we heat it to high levels so the 2 strands come apart. But the 2 strands are each radio-labeled so that when they find their complement and they're gonna find their complement in the clone that has the DNA from which that derives, okay? Then suddenly, they're going to bind to the DNA that came from that colony, for example. So all the radioactive probe will find its complement in that one colony. And after I've done that hybridization with that membrane, I wash the membrane, I put it under film, and the radioactivity will expose the film. I'll get this little spot of exposure. I'll see a black spot. It says, oh, there's a colony. If I now put this film on top of that plate of bacteria, I can identify where the spot is relative to where the colonies are. And I can say, oh, okay, so this colony must have a piece of the genome that includes 4.1. Okay? So, is that better? A little?
That are FLT though.
Okay.
I mean, it makes sense that you're taking a piece of G, inserting into a plasmid, checking where the plasmid is via film. But what exactly is an RFLT?
Okay. So a restriction fragment length polymorphism is exactly what it says. So when I said that I use this piece of DNA and I hybridize it to FLR1 digested DNA in one strain, and I got a 5 kilobase fragment, and then I use the same probe and hybridize it to another strain of fly, and I get a 3 kilobase fragment, then that is a fragment length polymorphism. 2 different forms, okay? Remember, polymorphism refers to multiple forms, right? And we were talking about green eyes versus brown eyes, or curly wings versus straight wings. Those are all polymorphisms associated with each with a single gene. This is a single gene where the polymorphism is in the restriction fragment length. Yeah.
Could an SNP result in a
Yes.
Okay.
So he's asking, can a single nucleotide polymorphism result in a restriction fragment length polymorphism? Yes, absolutely. So in fact, we often are more interested in those that do, because it's easy to screen for them. Okay? So in other words, I told you that FOR1 restriction enzyme site is GAATTC. If in one strain that site lies, specifically there's an FOR1 site up where 4.1 is, okay, and there is an FOR1. Of course, there are a bunch of FOR1 sites, right? But there could be an FOR1 recognition site, and then one of them, it still reads GAATTC, so FOR1 will cut there. But in another strain, it's been GAATCC, and now FOR1 will not recognize that. That's one nucleotide change, which gets rid of a restriction site. And so, naturally, if I have an FOR1 site, I hate not having a blackboard. If I have a site here, a site here, and a site here, okay? Then if all 3 are there, I'm going to get both this fragment and that fragment, right? This one gets mutated, I only get this big one, right? If my probe is hybridizing down here, when I have this site here, it's going to give me this size fragment, right? But if I've mutated that center one, when I use my probe, I get this much bigger fragment. And so that single nucleotide change, single nucleotide polymorphism, has led to a restriction fragment length polymorphism. Okay? So, I don't know that I really have, what I mainly want to tell you is that as soon as you find that one big clone that has the thing you're interested in, let's say it's, the one that has these things coming off of it. I do have a pointer, that much I do. Okay, say this is the one that hybridizes. You then can take pieces of this and hybridize to the rest of the library until you find, for example, this little piece will hybridize to this thing. So you pick up this second clone. So you see I'm just taking, I found this one because it hybridized to my 4.1, for example. All right? But I want to know more about this whole region of the chromosome. So maybe it hybridized and I can find it in the middle of this. But I'm going to take a little piece of DNA from one end of this clone and another from this other end of this clone. And I'm going to take those and use those as probes to go back and screen all these colonies again. And when I pick up another colony, what I'm picking up is either the same thing again, or I'm finding something that's right next to it. So, for example, in this picture, in this picture, if I use this as a little probe down here, I'm going to pick up this clone, and now I use this clone and take a piece of DNA from here, hybridize that to the whole library. I'll pick up this other clone, okay? And then when I use pieces of this, I can pick up that clone. And by this way, we end up putting the whole thing together. We get the whole DNA sequence for long periods of spaces, okay? Long spaces. So we can use just the DNA clones to stitch together to get the sequence for an entire chromosome. But then we use the RFLPs, the probes that we know see differences, to figure out where they are among those DNAs. Okay, so let me just quickly go to what we did before while you're still thinking with me. How did I find that 4.1 in the first place? It was totally random. Okay, so really what people have done is to just take out pieces of DNA and do these hybridizations, taking two strains they're interested in. So in this particular case, we had one kind of plant that's called the Columbia true breeding plant, and then the Landsberg true breeding plant that we knew were different for some traits we were interested in. So what we did is just take little pieces of DNA just at random out of the genome. And then we use them as probes to hybridize back to the genome of both this and that, cut with 20 different restriction enzymes. So we try it with FOR1 digested DNA from Columbia and Landsberg. We use HindU3 digested DNA from the two. We use XHO digested. And what we're looking for is a case where when we use our probe, it picks up one size fragment in Columbia and a different size fragment in Landsberg. That's our RFLP. Okay? So that gives us, that means that we can genetically follow that piece of DNA. Okay. Let me just quickly show you the southern technique so that I've got you with me. Remember I showed you this? When you digest genomic DNA from a complex organism like a plant and an animal, yeast, so forth, you're going to get, when you cut with BAMH1, for example, you're going to get a smear of DNA fragments, just a ton of fragments. It's a big genome. There are a lot of BAMH1 sites. You're going to get a lot of fragments. Nothing you can really do with that. If I looked at Landsberg versus Columbia, they look very similar when I cut them with BAMH1, and I just used rubidium bromide to detect all the DNA fragments in the gel. So this is a gel. You guys made pictures of gels, but you did it with nice little neat plasmid fragments. There are only a few fragments. If you take the whole genome, you get a smear like this, okay? So when I just told you, I'm now going to take some random piece of DNA, and I'm going to ask whether when I digest my DNA with BAMH1, or actually in this experiment, I think it was FLR1, we can look back, but I digested both Columbia and Landsberg with FLR1. They both gave bands that looked to be much like that, okay? A whole series of bands. But now I'm going to ask what size restriction fragment, what size FLR1 fragment is this random piece of DNA I've isolated? Okay, does it sit in? Okay. So to do that, I run my DNA out on a gel. I denature the DNA in place in the gel. Then I use, not terrible, I don't want you to know how to do a southern at this point. You're going to learn it in labs. But what you do is that you basically put your gel down on a surface, and you have very high salt solution that you force to go through the gel, and on top of the gel, you put this membrane. And so all of your DNA is being moved out of the gel onto this membrane, and all you see is this. Nothing. Okay? It just looks white. Actually, the rubidium bromide goes with the DNA, so it actually looks, you can see pink streaks, okay, from the rubidium bromide. So you have transferred all the DNA in a denatured form, meaning denatured, and this is, what I mean is the two strands have been pulled apart, okay? So they're not base paired to each other, but they're sitting right where they were. They really look just like this on this membrane, but you can't see it. And so you now cut a corner off this membrane so you know what's the upper right side, okay? And now you take that membrane and you put it into a solution. You first bake it as a way to make sure the DNA stays bound to the membrane. And now you incubate it in a solution with your denatured radioactive DNA probe that I just told you about, right? We can radio label the probe. The probe is some random piece of DNA we have. We've denatured it so it's not double-stranded anymore. And it's going to look for its complement among all those FLR1 fragments that are on that membrane. And in this example, I'm giving you a case where I incubated it with a probe. I then wash off all the excess probe, and then I take this membrane and put it under film. And now what you're looking at here is not the membrane, but the film. And for example, if my probe bound to a large not one fragment, I'll get one band there. This is one band for the BAM fragment. This is the size BAM fragment I'm getting, okay? So what I'm seeing is for this random piece of DNA I've picked up, it lies in the genome somewhere, okay? But it lies within a BAM fragment whose size I now know, and in a Bagel 2 fragment whose size I now know, and in an FLR1 fragment. And this is an example where now I would look not just at the Columbia DNA, but at the Landsberg DNA. And I'm interested in any random piece of DNA that gives me one size fragment with one enzyme, okay, say FLR1, in Columbia, but it gives me a different size fragment in Landsberg. Because that means I've got two alleles for that gene. Okay, yeah.
So you're using like one size, one radioactive marker, right? One size. But aren't those different sizes?
Oh, I'm so glad you asked me that. Because people get that confused a lot, okay? It does not matter how big your probe is. What matters is the size of the fragment in the gel. So I can have a long piece of DNA that's up there at the top of the gel. And my probe may hybridize to just one little part of it. But it's still going to pick up that big fragment because that's where that big fragment is. Okay? So the size you pick up has nothing to do with the size of your probe. The size fragment you pick up is the size fragment that DNA lies in the genome. So in this case, we've got it hybridizing to this size MOR1 fragment. If we go back to, and it looks like we're about finished, but let me just show you this. So this is an example of that, where when we use this one random piece of DNA, we picked up this size fragment in the Columbia DNA and that size fragment in the Lancet. And so then we can follow those as genetic markers. Okay, so I will post these slides on Blackboard.
This